Sure. Can you see my screen?
Well, I'll introduce you then. So, and I might not be able to do as good justice as Jake would. So we're so happy to welcome our speaker today, Dr. Ramana Davuluri, Ph.D. He's professor in the Department of Biomedical Informatics at Stony Brook University in New York. And he has, we have been together in many other contexts. I know Jake has worked with him. We were together faculty in the Department of Preventive Medicine, Division of Health and Biomedical Informatics at Northwestern University. That's where he was most recently. And we're happy to have him today, his talk, Application of Deep Learning Algorithms in Bioinformatics Methods.
Thank you, Amy. Thank you so much. First of all, my apologies for mixing up the time. Somehow I noted it as 12 Eastern. And then what happened was I was updating my computer and somehow I didn't open my email. So that's why I lost track of the time also. Anyway, thank you, everyone. So today I'm going to, I think, talk about some recent methods our group has developed. Some of it was developed by my students at Chicago, and then we're kind of continuing those after moving to Stony Brook. So our lab mainly works at the interface of gene regulation, particularly studying the gene regulation and the gene expression methods or informatic methods to study gene expression of gene regulation at the gene isoform level. Recently, we have developed a large language model for understanding gene regulatory code. So depending on the time, I will be talking mostly about this. Then a long-standing interest in our group is analysis of the cancer genomes, mainly for tumor subtyping. We have developed methods to subtype the cancer samples or tumor samples using the transcriptome primarily at the splice variant or isoform level. Recently, we have a DeepMOIS-MC, another deep learning based method that can perform integrative analysis of multiple omics data to derive robust subtypes that are associated with some outcome, for example, survival. We're also investigating the cancer drug and the drug target interactions, how they are affected by these splice variant protein isoforms. I'm not going to talk about that, but mostly I will be focusing on the DNA-BERT that we have released in 2021, which has become very popular and used in many applications now. And then time permitting, I'll get into the second project. So what do we mean by gene regulation and so gene regulation is a very like context dependent. In this cartoon, for example, in A, there is a promoter with the green arrow, and then there are several transcription factors that actually located in the promoter region that where the transcription factor proteins bind in a context dependent manner and drive the expression of the nearby gene. And of course, there are enhancers in the upstream region where there also the different transcription factors bind. So these are all, they don't act individually, they act in a coordinated and a team-based fashion. Those are the protein complexes we call in the biochemistry point of view. So then in a given promoter, if there are some variation, for example, SNPs are the mutations from the cancer point of view. Let's say in this cartoon B, there is a SNP that is A in the that is co-located in the TF binding site is changed to G, A to G. How is that affecting this the expression of the nearby gene? Is it going to produce more of the mRNA or less of the mRNA, which we which in the literature usually referred as EQTLs. So there is a lot of study in that, and there are EQTL analysis in the statistics literature that have been published extensively. Similarly, like these are genetic events, SNPs or mutations. There can be epigenetic events like methylation of the DNA. Now, how are they going to affect? We know methylated CpGs can silence the target promoters. So these are all these different functional sites we can look at in the genome. Apart from the protein coding regions, how are these affected by these genetic and epigenetic variants? It's been like so much of literature has been published on this, and there are many methods, informatic methods that are available. On the other hand, if you look at the right side, we are also very much interested in how are these producing different mRNAs. For example, this gene in this cartoon that has 4 exons can produce 3 different mRNAs that may give rise to 3 different protein isoforms. So one gene can give rise to 3 mRNAs and 3 different protein isoforms. Sometimes some of these may remain as non-coding, and some transcript variants can give rise to protein isoforms. So if we have the data, transcriptome or protein data, our lab mainly focuses on the transcriptome data and analyze the data at the isoform level or splice variant level or at the gene level. So making a matrices by gene by sample or transcript by sample and perform similar statistical analysis. How are the things going to change? Actually, we published quite a bit on that, and actually our conclusions vary. And it's kind of looking at old Google Maps versus the new Google Maps, I sometimes call like that. So you can zoom into deeper by analyzing the transcriptome at the splice variant or alternative splice variant level rather than at the gene level. Even from the biological point of view, what is the unit here? The unit that is made in the cell is an mRNA, a transcript, or eventually a protein. So what we measure should be attributed to the given transcript. If there are 3 transcripts that are made from a gene, maybe all 3 may not be expressed in all the cells. Maybe transcript 1 is expressed in cell type 1, whereas transcript 2 is expressed in cell type 2. But eventually, that is where we have to tag these values, expression estimates or expression values, whatever we call, to the units rather than treating the gene as a unit. So what is the context here? In this cartoon, which we have published as a review in the Transient Genetics Guide some time ago, there are 2 different core promoters, P1 and P2. P1, different protein, a complex of maybe 50 to 60 proteins combine and bind to that core promoter. P2, maybe a homologs of those proteins form a different complex and activate P2. Similarly, TF3, TF4 could be binding in the proximal promoter of P1, whereas TF1 and TF2 bind to proximal promoter of P2. So depending on how these complexes are formed, how they recognize different elements within those, that is what we call context. The context here could be cell type A or cell type B, or the sequence context, depending on TFBS1 or TFBS2, TFBS3 or TFBS4, that is the sequence context. So in terms of analyzing the data, if we are looking at, for example, ChIP-seq data, so we analyze the ChIP-seq data in cell type A, we may find a different set of peaks in the genome, whereas the cell type D, similar ChIP-seq data is available in cell type B, you may find different set of peaks. And now you take those peaks and analyze for the binding sites, you may get a different set of binding sites, a combination of binding sites. So that is what we mean by context. So why is it important from the cancer point of view? This again, a cartoon which we published some time ago. A given gene may have 2 different promoters. Promoter 1 could be a target of pathway X. Promoter 2 could be target of pathway Y. So one gene could be a target of different pathways, and these pathways could be switched depending on which promoters are used. And if we look at a recent publication of the pan-cancer transcriptome, 58% of the isoforms are results of usage of different promoters in cancer. So that's a lot. There's a lot of promoter switching is going on in the cancer. So now coming to the genetic variants at the SNPs, for example, here, if we look at the density of the SNPs in the entire genome and ask how are they located relative to the transcription start site? Panel A shows that all the variants, mostly the density is present near the core promoter region or the TSS. If we split that into SNPs and indels, you still see they are mostly present, co-located near the TSS. So what does it mean? I think the variation present in the promoters, obviously, what do they affect? They affect, for example, the core promoter elements like TATA initiator, BRE sites, and more in the proximal promoter region, transcription factor binding sites. So depending on how they affect these regulatory elements dictates how much of the corresponding transcript or the protein will be made in that particular individual, because we are talking about the population variation here. So with that biology background, what data we analyze? We data mostly analyze are the methods we developed to analyze the data are the RNA-seq at the transcriptome. Most of it so far we have applied in the bulk transcriptome. Again, I assume all of you know this. I'm not going to explain much about it. Similarly, the ENCODE data, the ENCODE consortium produces multiple data sets on multiple experiments. For example, there are 15,000 or more files that were conducted in 4,000 different experiments, looking at 767 different targets. The targets can be transcription factors or histone molecules. And then there are multiple cell lines. This data was produced. There could be primary cells or different tissues. So in terms of expression, there is a GTEx data. I'm sure all of you are familiar with this GTEx data in the normal tissues, expression in the normal tissues. And then from the cancer point of view, GDC data portal provides lots of the transcriptome and other data files, which we actually analyze using the methods we develop. So what we can do with all these data sets, we can obviously annotate these different regulatory elements, automated sequence annotation. We can get into mechanic insights, and we can prioritize which sites could be affecting these functional elements like promoters and binding sites. Using the splice variant information, we can come up with a better biomarkers or the signature of different cell types or different cancer subtypes that we use molecular subtyping or patient stratification. We can come up with better prognostic predictions, and we can also do time series and longitudinal analysis. Not going to talk much about protein isoforms, but I think that's where we are missing a lot, and a lot of drugs based on the analysis we published recently, a lot of cancer drugs do off-target effects because of these different protein isoforms. So the first one we talk about, this large language model which published in 2021 is DNABERT. This is a transformer based model to understand the DNA language in the non-coding genome. So you may ask, don't we understand the DNA language? Yeah, we can understand. We know what it is in the protein coding regions. Of course, there is another BERT model that was published later in 2022, but we can read a given protein sequence using this triplet codon table. But if we look at the non-coding region, we don't have anything like that. Usually, this is what we lead. There is some DNA sequence which we don't understand. I put these marks like the symbols. And then we say, this is an enhancer. And again, some sequence we don't know anything. And there is another enhancer. And then we continue. And then we may find something like GC something, maybe a P53 binding site or binding site of some other transcription factor, we can say. And then again, go on, and then say, here is the promoter, TSS, core promoter, and here where the gene begins. So this is mostly what is our understanding of a non-coding genome right now. So in comparison to the protein coding regions, this is what we have. So in the protein coding region, if there is a SNP or is a mutation, we can say, this is altering this amino acid. And if it is a non-synonymous, it could be deleterious, or it could be synonymous, depending on the table, amino acid table. It is not affecting the protein. Whereas in the non-coding region, if a SNP is overlapping with the binding site, we cannot really directly say whether this is affecting a binding site or not. Similarly, splice sites also in the intronic regions. So there is a tremendous need for having tools to understand the language in the non-coding regions. So with that context, we developed the DNABERT. And when we were designing we took the advantage of the natural language NLP context. So in NLP, there are two things. A one word may mean different things. For example, time flies like an arrow, fruit flies like banana. So there is a flies here, the word, but in the first sentence, the meaning is different, whereas in the second sentence, the meaning is different. So there is a context here. Similarly, in the DNA sequence, there could be a binding site that is present in one promoter, and the same binding site may be present in another promoter. We may do different functions depending on which other words or which other binding sites are present. Similarly, a meaning of a combination of three words may depend on a lot on the downstream words. For example, here is the man who blah, 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 important, did it. So the man who did it, there is a lot of explanation in between. It is like in the context of an enhancer. You have an enhancer way upstream, and it's actually driving the transcription or not driving the transcription of a nearby gene. So this kind of language understanding can be captured in a computational model. That is the main driving the thinking behind the development of or adopting a BERT model, which was already adapted in the NLP setting, or trying to understand the English language into DNA context. So this works like a pre-train and fine-tuning model point of view. So initial DNABERT was trained on the human genome. This is called pre-training. Basically captures the sequence composition into some kind of a transformer model. Once you have this pre-trained model, then you develop different fine-tuned models or different classifiers. It could be classifying promoters from the rest of the genome, classifying splice sites from the rest of the genome, which can also be applied to a different closely related species like mouse. So this is in a nutshell what is the DNABERT is doing. So it has only 4 letters. Unlike in English, preparing a dictionary of words is very tedious here. So previous programs, for example, the one I developed is a postdoc first TF or GenScan program used 6 MERS as words. So with that motivation, we took 6 MERS. Actually, we evaluated all the MERS, 2 MERS, 3 MERS, 4 MERS, and all. But we found out again that with multiple exercises, 6 MERS were the ones that are really working. So 6 MERS were the words here. So once you have these words, those are the tokenized, and then they get into positional embedding, and this becomes the input in the pre-trained model. So I'm not getting into the details of this. You can check the paper and the supplementary information if you are interested more in the actual model, how this was trained. So once we have that pre-trained model and the fine-tuned models, we perform DNABERT can do lots of downstream analysis. You can visualize important regions. For example, a sequence, you give it to DNABERT, and now the sequence is predicted as a promoter or not a promoter. So we focus on functional sites. Let's say it is predicted as a promoter. So if that sequence is predicted as a promoter, which of the regions within that sequence is very important to make the decision? Those are called high attention regions. So we can visualize, DNABERT helps to visualize those regions. Now, when we analyze ChIP-seq data, we take the multiple ChIP-seq files, peak files, and give it to DNABERT, and it can also do motif discovery. That is another module that was added here. And finally, most importantly, we can evaluate the effect of a variant. It could be a SNP that is in the dbSNP, or it can be a mutation on a transcription factor binding site, or core promoter, or splice sites. It can affect. For example, in this cartoon, the top panel has a that is mutated to C in the bottom panel. So that is affecting the function of that site. So in terms of now, because it's what DNABERT basically is doing, or the fine-tuned models are doing, is basically doing the classification or prediction. So the fine-tuned model DNABERT prompt effectively predicts the promoters, because when we eventually want to study whether a Tata promoter or the core promoter is promoter or not, it has to be highly accurate. So because we are studying the effect of one nucleotide or 2 or 3 nucleotides at a time. So the fact that one nucleotide can actually change the probability of a sequence of length 90 base pairs into a promoter or non-promoter, that's is like very challenging. So first of all, we want to make sure that the promoter is the predictive program is very highly accurate. So here this shows these RBC curves for Tata promoters and non-Tata promoters. Overall accuracy is very high, 0.98 to 0.93 for non-Tata promoters. Let's say you give a sequence here to this model and it predicts with that kind of accuracy as a promoter, and now you alter 1 or 2 nucleotides and give it again, and the promoter drops to 0.5, below 0.5, then those SNPs are the candidates that could be functionally affecting the promoter region or that particular promoter. So that's what eventually we will be analyzing. And the main intent of this program is not intended to predict promoters. We care whether there are a program to predict promoters or not. There are almost all the genome is annotated, but the main intent is studying the effect of the genetic variants or epigenetic alterations like DNA methylations. So here, the overall accuracy for the, similarly, we trained for all the TF transcription factors in the ENCODE data. So and we compared with other state-of-the-art methods like DeepMOIS, so and all, and DNA-BERT TF performed significantly better than other programs. Again, the accuracy is key here. If we don't have very highly accurate prediction program, it's then the effect of these 1 or 2 nucleotides studying that could be like we can't really evaluate the effect with the high accuracy. So this is how the DNABERT in terms of F1 score performs on the 690 ChIP-seq TF-based data, and it is 0.86 overall F1 score. Then there is also a module that you can visualize. For example, on the right side, you have a wiggle file from multiple sequences is a CTCF for ChIP-seq data. And in each line is a sequence here, or the CTCF bound region. And the bottom in that shows that the peak. And then each sequence, the blue line indicates a high attention region that makes the decision for that particular sequence to be a CTCF binding region or not. Now, if you take all those and put into motif finding algorithm, it actually predicts the CTCF binding site. So on the left side, there is a cartoon showing a TAP73 beta binding site. TAP73 is one of the long isoform of P73, a very important transcription factor in neuronal development. It's a dimer. See, this is the left one has a 0.44 and the right one has 0.51 using the position weight matrix scores. Whereas here the DNABERT predicting without taking any PWS into account actually is not a TF binding site as predicted like that. So DNABERT can take this kind of ChIP-seq data sets and then find the high attention regions in those regions and do a motif building, and which actually overlaps with the known motifs. So for example, here, this is a summary of all the ChIP-seq files. So after analyzing all the ENCODE data, we have unbiasedly, like without help of position weight matrices, unbiased motif finding using the ChIP-seq peaks. 1,595 were actually overlapped with the JASPER database. And there are 400 motifs that are undocumented. These could be false positive motifs, or this could be some novel motifs, which we need to investigate. So, but the fact that majority of these, without taking any help of position weight matrices or any known patterns, de novo finding these motifs tells that DNABERT motif algorithm actually is doing really a good job. So what do we have from DNABERT? DNABERT fine-tuned models can predict promoters of length -500 to 2,000, up to 2,000 base pairs. It can predict the core promoters, 70 base pairs around TSS, splice sites, length 30 by taking 30 plus or minus 60 base pairs. And then it can also predict TFBS binding sites and also build motif models. Right now, we are developing predictive models for enhancers and splicing factor binding sites. Okay, so then the important question is, what is the effect of a SNP or a mutation if it falls within the non-coding functional sites? So all these sites within that region, if a SNP or a group of SNPs fall in that, what is the effect on the prediction of that particular promoter or splice site? So this fine-tuned models probability, we can substitute the locus of interest with that mutation or that alteration. And we call that the substituted sequence as PS prime. So the genetic effect of the mutation BJ at locus I can be calculated using the predicted probabilities from DNABERT. So similarly, instead of replacing one base at a time, you can replace the entire sequence if we have the whole genome sequence from an individual. So we're currently working on that. I'm not going to show those results right now, but most of the results I show are using the DBSNP data. So then we calculate the score delta P, probabilities S prime minus probability S, and the multiplied by some score or log odds ratio you can calculate. And based on these two, we make the calls whether a SNP is affecting the site or not affecting. So this is a summary of the acceptor and donor sites. Overall, there are 295,397 acceptor and 297,957 donor sites. Then we like when we asked of these, how many have a SNP within that region, minus 32 plus 30. Most of these actually have at least one SNP. And out of these, 61,000 acceptors are affected by at least one SNP. Similarly, 72,000 donors are affected by at least one. Again, these are predictions and we have to validate it. But 20 to 25% of the functional sites are affected by at least one SNP with a significant change in the prediction probabilities. So similarly for Tata and non-Tata, also 24 to 31% are altered, Tata to non-Tata promoters. And these are three examples here. One is the CTCF binding site here, the first one. Second one is YYY binding site affected. And the third one is XPC, another CTCF binding site in XPC promoter. So all these are experimentally known that, but the SNPs are not known, but they're known to affect these binding sites. But these in the GWAS study, these SNPs have been implicated in the corresponding disease. So we are discovering these kind of functional sites linking to the disease. So right now we are in the process of analyzing the colorectal cancer data with a collaboration here at Stony Brook. Basically, the idea is to analyze the whole genome sequences and also using the VCF files. Similarly, we can apply this on the mouse ENCODE TFBS data as well. Although the model was primarily developed for human genome, we can apply it to the mouse genome as well. So when we applied, many of the TFBS sites in the mouse ENCODE data were actually accurately predicted. So can we use this to diagnostics? So we applied on the viral genomes. We actually developed a new model for viral sequence analysis. And then we tested the accuracy by just giving, let's say, give a sequence of length 200 and ask whether this belongs to any particular virus. And then we have seen that we need at least 2,000 bases or even 1,200 bases give rise to 0.95 accuracy. A 300 base pair region can give 90% accuracy. So it shows that DNABERT is capturing even closely related viral sequences and predict very accurately whether a given sequence can be predicted or accurately predicted into one virus or the other virus. Similarly, we tested for different strains, but predicting different strains of SARS-CoV-2 here, for example, because the changes are not a lot, but still with 1,000 base pairs, we can predict with 82% accuracy. So this shows that overall DNABERT is capturing and doing a very good job in predicting any sequence that could be different viruses, or it could be within one genome, one functional site versus other rest of the genomic sequences. So compared to other deep learning models, DNA models developing transferable understanding of DNA from unlabeled human genome that can generally solve various sequence-related tasks. So if we go back to the genome sequencing era, where I started my bioinformatics journey by developing gene prediction programs. So we used to have different programs for different sites. For example, I developed a program to predict promoters, and a lot of people have developed exons, predicting programs for exons, transcription factor binding sites, GenScan, like gene protein coding sequences, and so on and so on. But whereas here, DNABERT simultaneously can do all these things. It's like one hammer can take care of many nails in the in building the building, something like that. So it can globally capture contextual information from the entire input sequence with the attention mechanism. That is one of the main things that is helping this from the BERT model, helping understanding the DNA language here. It achieves a very high performance. It can uncover important subregions and potential relationships between different cis elements in the DNA sequence. It can also be adapted to closely related cross-organisms like mouse and monkey, all those genomes. We don't need to pre-train, because pre-training is a very tedious job, and it takes at least 20 days to 30 days, the pre-training step itself, depending on what kind of GPU cluster do you have. So future directions, it is very slow. Now we have actually a newer version. We are still testing it, but DNABERT is available and it's being used very well. So DNABERT 2 is recently released, and we are doing other benchmarking, particularly from the finding the SNP effects and cancer mutations. So this is what DNABERT is about. Amy, I think should I stop now? Because I think I passed the time.
Oh, you're 4 minutes over, which I think is actually pretty good. Like that's normal for anybody. And we still have quite a number of people who have stuck around, at least 20 people. So yeah, I didn't pull you out with the shepherd hook. So or throw tomatoes at you. So yeah.
So my apologies. I think I wasn't, because of my mistake, I'm not going to talk about the other deep learning method which we developed, but I'm going to stop now, and happy to take any questions.
Yeah, and yeah, thank you for everyone who has stuck around, and we understand if you have to go somewhere else because we're over time, but feel free to stick around. Sounds like Dr. Davuluri is around to answer any further questions. How much more were you going to cover in terms of your slides? Just kind of wondering where you were in your presentation, because you were going at a pretty good speed. I
was rushing a lot. Yeah, I wasn't explaining a lot of things, so I'm sorry about that.
That's okay. You give a taste, and they say speakers should always leave them wanting. You should always leave them wanting more. So okay, we have a question from Curtis Hendrickson, who is always looking forward to his great questions. I don't know if you can see the chat, or I can also read it to you.
Yeah, this is a question that apply bacteria or viruses that requires completely new pre-training. How long did the viral 28 sequence pre-training? Yeah, that it takes, depending for our computer, like the GPU cluster, it takes anywhere from 10 days to 31 month. But once the pre-training is done, the fine-tuning tasks won't take that much time. Did I answer my question? Any other questions you have?
So I had a question, but it's probably a fair way away from where you are. But I was just reading an article last night about the importance of ATP as a driver for everything that goes on in the cell. And the assumption has always been that the ATP level is substantial and adequate in all parts of the cell. And now people are beginning to realize under stress that's no longer true. So in the systems that you're studying, do you think that the availability of nucleotides to make messenger RNA, which is obviously going to drive the phenotype of that cell, becomes a limiting factor? And under normal cells, our cells don't divide very fast, but a cancer cell is quite different. So when you're under a very high stress situation, do you think this availability of the components to build the gene products is actually something that we have to consider? It's a mass action effect.
So if I understand this correctly, sorry. So the availability of the components, you mean like, can you?
Nucleotides or the transfer RNA, the assumption is that any of these reactions can be driven at an optimal speed because all the components for assembly are available and not limiting. And so under stress situations, and I use the cancer cell as a stress situation where the cells are dividing much more rapidly, does that then become a variable?
So if we have different situations and have the data available, I think we can build a model on that. One of the things we are right now thinking about is, right now, all the predictions are based on just the sequence.
Yeah.
So apart from the sequence, if we add other variables like the ChIP-seq data, like histone modifying data, and methylation data as other layers of data, and then use the fine tuning to predict one condition versus another condition, the sequence and other experimental data could predict different situations like this. That is something I think can be doable if the data is available.
Sure. Okay.
I hope I understood your question properly.
Yeah, I think you did a good job. Thank you.
Thank you so much.
Also, just a quick note, I put the links in the chat for the hackathon registration and also for the prep session. So we have a hackathon tomorrow, and on patient safety, AI, ML, Ramana, and I kind of like stuck you on there as part of our pre-hackathon. Even though it's like not a bioinformatics thing, it's still like, using AI, ML, data science techniques. So hope you don't mind that. So there's another question from Curtis. Is there manuscript published for the DNA-BERT for NGS-based DX, specifically the viral DNA-BERT model?
This is we just recently submitted. But you are interested, we are happy to share that model.
Any final questions? Well, hearing none, feel free to contact Dr. Davuluri if you have any further questions. I can also have his, okay, Curtis, I'll send you that. I can send that to you, Ramana, that thank you for the presentation. I appreciate, I think in informatics, we have this attitude of, making it work no matter what. And, we do what we can. So thank you for the enlightening presentation. Thank you everybody for attending and staying till the bitter end. And have a wonderful weekend. Hope to see you at the next Power Talk.
Thank you, Amy. Thank you so much. Thank you, everyone.
Yep.
Have a wonderful weekend.
Yeah. Bye-bye.
Bye.